Welcome to Quick MCAT Lessons, where I try to explain MCAT topics in 10 minutes or less. Today, we are continuing our discussion on the cell cycle and we'll go over the mitotic phase and cell cycle regulation. After the cell passes the G2M checkpoint or the G2 checkpoint, it enters the M phase or mitotic phase. This is the stage of the cell cycle in which a cell divides its duplicated genetic material and cytoplasm to form two genetically identical daughter cells. This is a tightly regulated and dynamic process that includes mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm. Mitosis itself is divided into several stages: prophase, metaphase, anaphase, and telophase. A mnemonic I use to remember the phases of mitosis is PMAT, which is prophase, metaphase, anaphase, telophase. And you will see that meiosis is just PMAT times 2, because there's prophase 1, prophase 2, metaphase 1, metaphase 2, etc. So just as a quick reminder, mitosis occurs in somatic cells, which are cells that are not involved in sexual reproduction, and meiosis occurs in germ cells, which are cells that are involved in reproduction. So let's start with prophase. Prophase marks the beginning of mitosis. During this stage, chromatin condenses into visible chromosomes, each consisting of two sister chromatids joined at a centromere. That nuclear envelope begins to break down, and the centrosomes, which have duplicated during interphase, migrate to opposite poles of the cell. These centrosomes begin to form the mitotic spindle. The mitotic spindle is a structure made of microtubules that plays a crucial role in organizing and separating chromosomes during mitosis. It is anchored at opposite poles of the cell by centrosomes. Spindle fibers extend from centrosomes, and these spindle fibers are made of microtubules. They attach to the kinetochores of chromosomes to align them at metaphase plate, and then pull sister chromatids apart during anaphase. Surrounding each centrosome are asters. These are star-shaped arrays of microtubules that help stabilize the spindle and position it correctly within the cell. So overall, the mitotic spindle, spindle fibers, and asters are all composed of microtubules. They just do slightly different functions, but the overall function is to ensure proper chromosome splitting. So in prophase, the nuclear membrane dissolves, allowing the spindle fibers to contact the chromosomes. The nucleoli becomes less distinct and may even disappear. Kinetochores appear at the centromere. Kinetochores are protein structures located on the centromeres and are the attachment site of kinetochore fibers. And so in metaphase, the spindle fibers attach to the kinetochores. The chromosomes are aligned along the metaphase plate, which is an imaginary plane equidistant from the two poles of the cell, which essentially mean it's at an equal distance from both poles. This precise alignment ensures equal distribution of genetic material. After this, we have anaphase. Anaphase begins once all chromosomes are properly attached and tension is sensed at the kinetochores. The centromeres split and the spindle fibers shorten. This pulls the sister chromatids apart and toward the opposite poles of the cell. Each chromatid is now considered an independent chromosome. Finally, during telophase, the separated chromosomes reach the opposite poles and begin to decondense back into chromatin. Telophase is essentially the reverse of prophase. The spindle disappears and a new nuclear envelope forms around each set of chromosomes, effectively re-establishing two distinct nuclei within the cell, and nucleoli reappear during telophase. Finally, cytokinesis occurs. This is not technically part of mitosis, but it completes the M phase. In animal cells, a cleavage furrow forms due to actin myosin interactions, pinching the cell in two. In plant cells, a cell plate forms due to vesicle fusion, eventually developing into a new cell wall. The result is two generally identical daughter cells, each ready to enter the G1 phase of interphase. The entire M phase is closely monitored by the spindle assembly checkpoint, which ensures that the chromosomes are properly attached to the spindle before anaphase begins. Failure in this checkpoint can result in aneuploidy, which means an abnormal number of chromosomes in a cell, which is a hallmark of many cancers and other diseases. Now let's talk about regulation. So the cell cycle is regulated by a coordinated network of proteins that ensure that each phase occurs in the correct order, and each phase only occurs when conditions are appropriate. This regulation maintains cellular homeostasis and prevents errors in DNA replication and cell division. The primary regulators include cyclins, cyclin-dependent kinases, which are called CDKs, cyclin-dependent kinase inhibitors, or CDK inhibitors, and various cell cycle checkpoints. Cyclins are proteins whose concentrations fluctuate throughout the cell cycle. They bind to and activate CDKs, which are enzymes that phosphorylate specific target proteins to drive the cell through different stages. Each cyclin CDK pair controls a distinct transition. So you don't need to know exactly which cyclin controls what, but I will tell you them nonetheless. So cyclin D and CDK4 and CDK6 are active during G1. They help the cell grow and prepare for DNA synthesis. Cyclin E and CDK2 help the cell get past the G1 checkpoint, or past the G1S transition into S phase. After it's in S phase, cyclin A and CDK2 take over, and these function during the S phase to ensure DNA replication proceeds. Finally, we have cyclin B and CDK1, which control the G2 and M transition and entry into mitosis. Now, cyclin CDK activity is tightly regulated. One level of control involves CDK inhibitors, or CDKIs, such as P21, P27, and P16. These can bind to cyclin CDK complexes and block their function. These inhibitors often act in response to stress signals or incomplete cell cycle events. The main control protein is P53, and this regulates P21 expression. Now, the cell cycle also contains checkpoints, which serve as control mechanisms. We talked about these checkpoints before, but I will just go over them really quickly. So the G1S checkpoint ensures that the cell is large enough, has sufficient nutrients, and has undamaged DNA before committing to DNA replication. The G2M checkpoint verifies that DNA has been fully replicated and is undamaged before the cell proceeds to mitosis. The M checkpoint, or the spindle checkpoint, ensures that all chromosomes are properly attached to the mitotic spindle before proceeding to anaphase. Another key player is the retinoblastoma protein, or RB. In its unphosphorylated state, RB binds to the transcription factor E2F. This inhibits transcription of S-phase genes. But when cyclin D with CDK4 or CDK6 phosphorylates RB, it releases E2F. This allows transcription of S-phase genes and allows the cell to proceed into S-phase. This step is crucial for controlling the G1S transition and is frequently mutated in cancers. Now, together, these regulatory mechanisms ensure the fidelity and timing of cell division, allowing cells to divide only under the right condition and in a controlled manner. That's it for today. Next time we'll talk about cancer.